Obviously, cytotoxic T cells should not attack the beta cells of the pancreas. What should happen is in the thymus, cells should express the same proteins that will be expressed by the beta cells of the pancreas, and any cytotoxic T cells which recognize these as foreign are inhibited by Treg cells. If, however, there is a mutation in the gene AIR, which causes the thymus not to express the peptides of the periphery, then cytotoxic T cells might never get the chance to react against them or be selected against. There could be mutations in the MHC1 complex, the HLA class 1 genes, which would interfere with the presentation of peptides like insulin or a portion of it to screen out self-reactive cytotoxic T cells. Or the Treg cells might not inhibit self-reacting cells. There should be no helper T cells which would react against the peptides of the pancreas and promote then the cytotoxic T cells or the B lymphocytes which would do the same. The reason that this should not occur is that if the thymus expresses the peptides of the periphery, including the pancreas, then antigen-presenting cells, such as dendritic cells, macrophages, and B cells can take up these peptides and have them bind to MHC2 proteins and present them to T helper cells. Any T helper cells which react against self could be inhibited by Treg cells. If, however, the thymus is not expressing genes from the periphery because of mutations in the gene AIR, if there are mutations in the HLA class 2 genes which interfere with the presentation of insulin peptides, these mutations are among the most common which cause diabetes, or mutations in the Treg cells, then this buildup of tolerance might not occur. B lymphocytes should not produce antibodies which react against self. This should be managed through both central tolerance in the bone marrow, by which any self-reacting B lymphocyte does not leave the bone marrow for the periphery. And this can be accomplished through clonal depletion, where programmed cell death or apoptosis is triggered in self-reacting B cells, in anergy, where simply self-reacting B cells are not given the stimuli to survive, or even B receptor editing, where the antibodies are actually reshuffled so that they bind a different protein. Tolerance can also be built in the periphery in regions such as the spleen and lymph nodes through anergy, the desensitization of receptors, and the expression of inhibitory co-receptors. Obviously, these mechanisms must sometimes fail, given that antibodies against proteins of the beta cells in the islets of Langerhans, such as antibodies against insulin, not only form, but are one of the earliest precursors to diabetes. And the transition to preclinical diabetes is the generation of antibodies against two or more self proteins.